Chapter 3 deals with the cells, which is the most basic fundamental unit of life. So we're going to look at all the components of the cells and different processes that are continuously going on in the cell. Robert Hooke, from a historical standpoint, was the first one to see a cell back in 1665. He was looking at cork, noticed a very consistent looking arrangement too. And then Antoine van Leeuwenhoek in the late 1600s, he is known as the father of microbiology. He did a lot of studies looking at various samples and made his own microscopes. The cell theory just states that the cell is that basic fundamental unit of life. An organism's activity is dependent upon the various activities that are going on within each individual cell, and then how the cells are going to be working together to carry out additional activities. The activities that are occurring in the cell from a chemical and biochemical standpoint are determined by the structure. Structure determines function even when you're looking at the cellular level. And one of the most basic things of the cell theory states that cells only arise from other cells. They don't magically appear, they have to come from other cells. So cells have different structures, they have then different looks to them. So you can have cells that help to connect one part of the body to another part. They can line different membranes, so different openings, different cavities. So in the human body, we've got things like this, such as erythrocytes, or your red blood cells. You've got epithelial cells, such as here, and this fibroblasts are in your connective tissue. You have cells that will store nutrients, such as your fat cells here. You have cells involved with the immune system. They recognize foreign substances in the body and engulf and destroy them. That would be a macrophage. You have cells for reproduction. You have some cells which are able to contract and whose purpose is to provide movement. And then you have some cells, such as a nerve cell, whose purpose is to gather information and help to control ultimately different functions. In terms of sizes of cells, they come in all different shapes and sizes. You do have things such as, on the scale here, some cells that you can see with the unaided eye. The chicken egg is considered a cell. Frog eggs, you can often see those with the unaided eye. With a light microscope, this is where most of the cells are that we are going to be looking at in this class. Plant cells tend to be larger than animal cells. Both plant and animal cells are much larger than, say, bacteria, which in turn are much larger than viruses. Viruses are way down in this area where you must use an electron microscope. So the cells do come in all shapes and sizes. Now, first we're going to look at some of the extracellular materials. These are materials outside of the cell. We have various body fluids. Ronaldo saw there's a lot of fluid. About 60% of the body mass is water. These body fluids are divided into different parts or different components. You have the interstitial fluid. This is fluid that's between the cells. You have cerebrospinal fluid, which is in the brain and also the spinal cord, helping to provide fluid. It helps to lubricate. It should not have any type of, certainly, foreign substance in it like bacteria. And then blood plasma is another one of the body fluids. Cellular secretions, these often assist with lubrication and digestion. Sometimes there are components that you may engulf that are too large to go across the cell membrane, and so the cell has to secrete different enzymes to break it from a very large compound into smaller components that then can be brought inside the cell. This extracellular matrix, commonly referred to as your cell glue, helps to hold everything together. Now, there's two basic categories of cells, eukaryotic cells and prokaryotic cells. animal cells are eukaryotic cells, so those are the ones that we're going to be looking at. They tend to be much more complex and larger than prokaryotic cells. One distinguishing feature of eukaryotic cells versus prokaryotes is that a eukaryotic cell does contain membrane-bound organelles. Those organelles will have very specific functions, and once again, it all works together for the overall functioning of the cell. Another distinguishing feature of eukaryotic cells is it does contain a nucleus. Now, all cells will have DNA. It just depends on where is it. In a eukaryotic cell, the DNA is inside the nucleus, which is inside the cell. So it has some extra protection. The DNA is separated from the cytoplasm. So in this representative cell of an animal cell, you can see in the very middle here, we do have the nucleus. This is where the DNA is enclosed in here. It is membrane bound, so there are these little pores, these little openings that allow materials to flow from the cytoplasm, which is outside the cell, into the nucleus, and things from the nucleus to move out through these pores. Inside the nucleus is the nucleolus, and this plays a role. It does produce ribosomal RNA, which is going to be needed later for protein synthesis. Chromosomes, that's your DNA. Now, there's a lot of organelles, as I said, outside of the nucleus. As you can see here is the smooth endoplasmic reticulum, the rough endoplasmic reticulum, the Golgi apparatus, the mitochondria. You have these other vesicles, some of them peroxisomes, some lysosomes, the centrioles, which are going to be used for cell division. And notice there is this outer membrane, that is your plasma membrane or your cell membrane, that separates the internal components of the cell from the external environment. So all cells will have a plasma membrane. Some cells will have an additional cell wall on the outside of the membrane, such as plant cells, fungal cells, bacterial cells, but animal cells, your cells, do not. So we're going to look at all these various components of the cell. So in a very broad division, the structure of the cell, you've got that plasma membrane, you've got the cytoplasm, and then you have the nucleus. Membranes are going to help control what's able to move in and out of either the cell, or if you're looking at the membranes on a specific organelle, it controls what moves in and out of that organelle. What allows it to have a very specific function is directly related to the type of proteins that are embedded in that membrane or on the surface of the membrane. Now, membranes are described as having a phospholipid bilayer with these proteins that are embedded in them. It's also described as having a fluid mosaic model, which just means that molecules can kind of drift in and out of the membrane. There's some side-to-side motion in there as well, kind of like seaweed drifting back and forth. It's selectively semi-permeable, meaning only certain things are going to be allowed to pass through. Cholesterol is a compound that helps to stabilize the membrane, so it's necessary that you do have some cholesterol. This is a schematic drawing of membrane. In this case, it is a plasma membrane or cell membrane. If you notice, here are your phospholipids, and it's a bilayer, so it's two layers of those phospholipids. If you remember from the chemistry, the tails are hydrophobic. They don't like each other, so they're going to point in towards each other. The heads are hydrophilic, meaning they do react with water, so they're going to be on the external surface of the membrane. So one side's pointing in the cell, one's pointing out of the cell if this is a cell membrane. All of these kind of purplish structures here, these are all proteins that are, as I said, embedded in the membrane. And these smaller little yellow structures right here, that is cholesterol that's helping to stabilize. So when we say it's selectively permeable, once again, only certain things can move across the membrane. And when we say it's a fluid mosaic, it just means there's some side-to-side motion here. And once again, this is just another depiction of it. Hydrophobic, hydro means water, phobic is fear, so remember that means the center portion here is hydrophobic, and that's going to play a big role in helping to determine what can pass through and what cannot. If something is readily dissolved in water, it's going to have problems passing through the hydrophobic section. So there's lots of different types of cells and different characteristics of these different types of cells. Some of that's going to be determined by, once again, the membrane proteins. And within a cell, some of the organelles have membrane, they're membrane-bound organelles. So their membrane is also this phospholipid bilayer, and the proteins that are embedded in those often give the very unique characteristics for that particular organelle. Another thing I want to point out here is this is on the inside of the cell down here. And so you'll notice that we have the cytoskeleton filaments here, helping to stabilize everything. Once again, cholesterol here. An integrin is a protein that does extend the entire width of the membrane. And then you have submembranes that are, or proteins, excuse me, that are extending out on the outer surface of the membrane. A glycoproteins right here. That just means it has a sugar attached to the protein. Here you've got a sugar attached to a lipid. And this is the extracellular matrix. When I mentioned that you've got materials outside the cell, that's what all of this is. So what types of proteins may be embedded in the membranes? Well, you have lots of different types. You can have integrins, glycoproteins, junction-forming proteins, enzymes, receptors, and proteins involved with transport. So we'll start with the integrins. As I just said, they span the entire width of the membrane. So to hold them in place, they will be attached to the cytoskeleton on the inside, and then on the outside of the cell, they're actually attached to the extracellular matrix. So like I said, that's going to help hold it in place. It helps to give strength to that framework. Glycoproteins. So this has sugar attached to a protein. This is very important for cell-to-cell recognition. So the outer part that sticks outside the cell often has carbohydrates. What that carbohydrate is, it depends on the cell, it depends on the individual, it depends on the species. So there can be quite a bit of variability there. Glycoproteins are really important because it allows for the cell-to-cell recognition. It's very important in your immune system and allowing your cells to be able to recognize that some of these are unique to you, so mine are different from yours. And that's what allows what we call self versus non-self, that your immune system is able to recognize, okay, all of these cells are part of polyp. And so that's self. Now, if there were to be an introduced foreign cell, it would recognize some of these glycoproteins saying, we haven't seen this before. This is a part of polyp. So that is non-self, and they are programmed to basically seek out and destroy that. It doesn't distinguish between, oh, that is some pine pollen, which isn't going to kill her, versus, oh, this is a bacteria, this is salmonella, which could make her really sick. It just sees it as non-self, and it will attack and destroy it. And you'll study a lot more about how that works when AMP2 you study the immune system, but just know that it has to do with these glycoproteins. Junction-forming proteins. There are in animal cells 3 main types of junctions, and they're formed by proteins. There are tight junctions, anchoring junctions, and gap junctions. The tight junctions help to form a seal around the cell, which helps to prevent leaking of extracellular fluid. You don't want to have this fluid loss. Anchoring junctions, also known as desmosomes, help fasten the cells together. They're kind of like rivets, if you want to think of that. And then the gap or the communication junctions, these are pores between cells. cells. And this picture depicts the different types of them. So you've got your tight junctions. Here's those anchoring, like I said, they're kind of like rivets holding the cells together. And then the gap junctions are literally pores. So you can have communication, essentially, between adjacent cells. Enzymes. Enzymes are a class of proteins or a group of proteins. They act as a catalyst. That means we've talked a little bit about them previously, how they will speed up the rate of a reaction, excuse me, without being used up itself in that reaction. Oftentimes in a chemical reaction, each step has its own specific enzyme. So they have to oftentimes work together and in a specific order. So oftentimes what you're trying to do is you have your first substrate, it's going to bind to an enzyme and it produces this product, which now becomes a substrate for a second enzyme, and then it produces the final product. But this has to occur in this order. And so we'll see that a lot with specific chemical reactions. Enzymes tend to be very, very specific for which substrate they will act on. Receptors, these are proteins. They typically think of them as like a messenger. They're receiving the message from another cell and then will communicate that message now to the internal part of the cell that it bounds to. It has a very special shape to it that's unique to that message. It's kind of like a docking center, if you will. So here comes the message. Here's your receptor protein. So it's receiving the message, and then it in turn acts kind of like a message to say, okay, we need to activate something inside the cell. One way of looking at this is to keep in mind that whenever you have, in this case, a messenger comes in, this is going to be very specific that it will bind in this spot. But whenever you have binding, you have a shape change. So when the message right here binds to the receptor, it's going to cause this shape change of that receptor, which then activates this, which causes a shape change, which activates this, which causes a shape change, and then finally activates your final molecule. So once again, it has to occur in a certain sequence. But that receptor, see how it's sticking out, protruding some on the external surface of the membrane so that it can bind to this molecule. Transports. A lot of ions that are soluble in water, therefore will not be soluble in lipid. You're either soluble in water or you're soluble in lipids. Water and fats don't mix. So if you can't mix with the lipid, which is what the membrane is composed of, those phospholipid bilayers, if you can't mix with that, you're going to have problems, as I said earlier, crossing that membrane. So how are you going to get across? Well, oftentimes you may need help from transport proteins. And so there's different types of transport proteins. Some of them are like this protein that's here on the left, where there's actually a pore through the middle of it. Think of it like somebody stuck a straw through the membrane, and now in the middle here, this little open channel, that is hydrophilic, so these substances can flow through. Some of them, instead of being a continuous open like channel, it's what we call a gate. And so this is still the protein. You have your substance come in, it's going to bind once again, and that binding then a shape change, and it's like it opens a gate. Now this oftentimes requires ATP, which is energy. So there's different types of transport, what we call passive transport versus active transport. The difference between them is that passive transport does not require energy. Why? Because your substance is what we say moving down its concentration gradient. Well, great, what does that mean? It just means it's moving from an area of high concentration to an area of low concentration. A gradient means you have different concentrations. Which way is it going to move? From a chemical standpoint, things always want to go from high concentration to low concentration. That's the way it naturally wants to go. So you don't have to put any energy into it for this to happen. Now, active transport does require energy. Why? Because you're going against that concentration gradient. You're trying to go from an area of low concentration to an area of high concentration. It doesn't want to go that way. So that concentration gradient, remember you're talking about the concentration or the density of a chemical in a certain area. Now with passive transport, there's three main types that we see in biology: diffusion, osmosis, and facilitated diffusion. Diffusion, because it's passive, it doesn't require any energy, and you are simply moving things from an area of high concentration to a low concentration, and this will continue until you reach equilibrium. So this is showing here, and you can try this at home if you want. If you just take a glass or whatever container you have and put water in it, and then put 1 drop of food coloring. Just let it sit, don't mix it up. Well, the drop has a lot of the dye in it, right? The rest of the water doesn't. So here's all this dye. Just leave it sit. Now this is showing with a membrane to simulate a cell that you would have this membrane. In purposes of doing this in your home, like I say, just use a glass of water and some food coloring. 1 drop. And what happens? Just let it sit. And notice that the drops are moving from this left-hand side over to the right-hand side. Why? Because it's high concentration on the left, low concentration on the right. So it's naturally going to want to move until you reach equilibrium. So your food dye in your water, do you have to stir it up? Do you have to heat it up? No. If you just, well, if you're impatient, you might want to stir it up. But, for purposes of this, just let it sit and come back to it later. And you'll notice that it will eventually spread until it reaches equilibrium. That's all diffusion is. If you have 2 or more substances, each one will diffuse down its own concentration gradient, independent of each other. And one of the best examples in the human body is in the lungs. When you inhale, atmospheric air is very high in oxygen, low in carbon dioxide. So when that atmospheric air goes down into your lungs and you have a lot of blood supply to your lungs, that blood has been throughout the body. It's already kind of been used in terms of the gases in it. And so in the blood, as it enters the lungs, it is very high in carbon dioxide concentration, very low in oxygen. Well, gases, just like liquids, want to go down flow down their concentration gradient. They're going to diffuse. It's not just liquids, it's also gases. So the atmospheric concentration of gases, the oxygen is very high. It's very low in the blood. So the oxygen is going to flow from high to low, so it flows from the lungs into the blood. Now the carbon dioxide was high in the blood but low in the lungs, so it's going to flow the opposite direction. They don't care. The oxygen doesn't care what the carbon dioxide is doing or vice versa. They both just go down their own concentration gradient, and it's kind of like, hey, the oxygen, I'm flowing down my concentration gradient. Carbon dioxide, you're on your own. Do your own thing. I don't care what's happening to you. I'm just worried about me. And so when you have 2 or more substances, they will diffuse down their own concentration gradients. As that blood then leaves the lungs, now it's high in oxygen, low in CO2, which is carbon dioxide, and it's going to flow back to the lungs and then to the rest of the body. Now when it gets around to the rest of the body, which needs oxygen, the oxygen in the cells is low, high in blood, so now the oxygen is going to flow out of the blood. Carbon dioxide, which is a waste product of a lot of chemical reactions that are occurring, tends to build up high in the cell, and so it's going to be higher in the cell, low in the blood. Carbon dioxide goes back into the blood now. So now the blood around the cells has now, you've had this exchange occur throughout the body, the tissue level, and as the blood comes back to the lungs, now it's changed and it's now low in oxygen, high in carbon dioxide, goes back to the lungs, have the exchange occur there, and this just keeps repeating. So this is showing this essentially with these dyes. So you have the green dye, you have the purple dyes. Once again, each one is going to diffuse according to its own gradient, ultimately trying to reach equilibrium. Now, polar molecules and ions can diffuse across the plasma membrane oftentimes if they have a transport protein that can help them. And this all goes back to the membrane being hydrophobic. Typically, polar molecules tend to be hydrophilic. They will dissolve in water, and so they're not going to like going across that membrane. So facilitated diffusion, really, the way to think of it is it's the same thing as diffusion. You're going from an area of high concentration to low concentration. So you're flowing down your concentration gradient. It's a type of passive transport, so no energy is required. How are you doing it? What's the difference in? Well, facilitated diffusion just means you need some help. You need a little bit of help from whom? From one of these transport proteins. It may have to provide a tunnel for the passage. It may have to bind and then be released on the other side. But somehow there is this protein that is going to be assisting these polar molecules and ions to get across the hydrophobic area. So if it can move by itself, it's diffusion. If it needs help with the protein, then it's facilitated diffusion. So what are we talking about? Oftentimes sugars, some of your amino acids, ions, and sometimes water itself. Now, there are some cells that have special transport proteins that are known as aquaporins. These help make the transport of water occur very quickly. These are in cells that tend to have a high concentration or a high need for water. Things such as plant cells, which don't concern us too much in this class, but in terms of the human body, certainly kidney cells and red blood cells have these aquaporins because they're transporting so much water. Now, osmosis is still a type of passive transport, so no energy is required. Essentially, with osmosis, what you are talking about is diffusion of water. You're going to be moving the water down its concentration gradient. Now, something just to keep in mind is that equal concentration does not necessarily mean equal volume. So in this diagram here, you have over here on the left, you have this depiction of with a membrane right here in the middle, and you've got something dissolved in the water. The solute is what is dissolved in the water. Now right here you can see it's equal volume, but that doesn't mean equal concentration. So when you get equal concentration of the solute, you now have a difference in volume. It's not the same volume. So just be aware of that. And this, we use the term tonicity, is referring to a solution that's able to either gain or lose water. And there's three different terms that we use: isotonic, hypotonic, and hypertonic. Use your vocabulary to help you. Iso means the same or equal. Hypo means less. Hyper means more. And you are doing a comparison of the solution to what's inside the cell. So if you're talking about an isotonic solution, that means the concentration of the solute, of what's dissolved in it, because that's what you're looking at. It's kind of a two-step process. You're going to look at what is the concentration of what's dissolved in the water, inside the cell as compared to the solution. So you're doing a comparison between those two things. So you want to see what's dissolved in the water, that's solute. Is it the same inside the cell and the solution? And usually what you're going to be doing is looking at the solution as compared to the cell. Is the solution higher than what the cell inside the cell, or is the solution less than what the... Now, once you've determined that, then you can figure out which way is the water moving. Is the water going to be flowing into the cell or out of the cell, or is it going to be equal? Now, with the isotonic solution, because the solute is the same inside the cell versus outside, inside and outside is the same, the volume is going to remain constant. This is a normal, healthy situation for animal cells. For plant cells, the plant's going to be limp. For humans, the isotonic solution is 0.89% sodium chloride. That is what saline solution is. Now, just to give you some practical applications of this, if you wear contacts, you know when you first got the contacts, you were told to rinse the contacts with saline solution. Now I'm talking about what you were told when you first got them, and I'm not talking about what you may actually do, because we know a lot of times people kind of forget about that. Why were you told to rinse them in saline solution and not just tap water? Well, saline solution is isotonic, so it's equal to what your cells are. You're taking your contact and you are putting that right against your eye. You don't want it to end up drying your eyes out. And so you want it to be equal. That's why you're using a saline solution. Some of you have medical experience already. Some of you do not, which is fine. But all of you at some point or another have watched, if nothing else, a movie or a TV show. Doesn't matter which one it is. You've had some, either a catastrophe or maybe it's just an accident or someone's sick. Anyway, they're wheeling a patient into the ER and everyone's rushing about. And what's one of the most common things you're going to hear them say? Start an IV of saline. Why? Well, obviously you're dealing with some other issues. You don't want to make it worse. So saline solution is equal to what your cells is the normal situation for animal cells, which of course we are. So now what about a hypotonic solution? Hypo means less. So this means the solute concentration outside the cell is less than what's inside the cell. And what's going to happen is the water wants to move towards wherever the higher solute concentration is. So if it's higher inside the cell, the water is going to move inside the cell. It's less outside, all that water then is going to move inside the cell. So what happens to animal cells? Well, you've got the water moving in and it's trying to reach equilibrium and it keeps moving in. Well, what happens to that poor cell? It starts swelling up and it can only swell so much. It's like when you blow a balloon up. It can only get so much bigger before eventually it's going to burst. For plant cells, because they have a cell wall, it keeps the cell from bursting. And this is actually a healthy state for a plant. A hypertonic solution means that the solute concentration outside of the cell is greater than what's inside the cell. So the water is going to move towards that higher concentration, which is outside the cell, so that means the water is flowing out of the cell. And what happens? The water is flowing out. It keeps flowing out, trying to reach equilibrium, and the cell's losing water. It's going to shrivel up. Plant cells, same type thing. The water's going to shrivel up, and that's when the plant starts to wilt. So this diagram is showing plant cells on the bottom, animal cells on top. As I said, isotonic solution, this is the normal situation, nice healthy cell. Hypotonic solution, the cell's going to burst or lyse. And then the hypertonic solution is going to shrivel or crenate, is the term we use. Osmoregulation, this controls the amount of water uptake that's necessary for animals to survive when they are in either hypertonic or hypotonic solutions. Now, active transport does require energy because you're moving against that concentration gradient. If we say it requires energy, you should be thinking ATP because that's where usually the energy is going to come from. So why would you expend energy to move something where it doesn't want to go? And the reason for this is that it does allow a cell to maintain a concentration gradient. One of the best examples are with the sodium and potassium. Under most conditions with cells, the level of concentration of your potassium is higher inside the cell than outside. And then sodium typically is going to have higher concentration outside the cell and low inside. So with diffusion, what's going to happen is your potassium that's high inside a cell is trying to move out. The sodium that's high outside is trying to move in because it's trying to reach equilibrium. And you're like, okay, so that's great. Why can't you let it reach equilibrium? Here's the problem. In order to have your nerve impulse be generated and sent down from nerve to nerve, and then for one of those nerve cells to stimulate your muscles in order to have muscle contraction, these electrical impulses, what has to happen is that you must have high potassium inside the cell, low sodium inside the cell. So you need to keep potassium in and sodium out. You have to have this concentration gradient. If you do not maintain this concentration gradient, you would never generate electrical impulse, you would never generate muscle contraction, and basically then you would die. So you must maintain this concentration gradient. You cannot let it reach equilibrium. And so what happens is the cells have what we call a sodium-potassium pump, whose main purpose is to help keep this gradient. You maintain this concentration gradient. We have studied this extensively. We know everything about it and how it works. And so the sodium-potassium pump, basically it's an enzyme. And what it's going to do is as the sodium is trying to flow in, diffuse into the cell, it's going to pump it out. And the potassium that's diffusing or leaking out of the cell into the environment outside the cell, it's going to pump it back inside. So this is in all of your plasma membranes, the high concentrations in your nerves and your muscles where you have to maintain this gradient. And so an antiporter just means it's a protein, transport protein, and it's pumping two things in opposite directions. So it's going to be pumping sodium out and potassium back in. Because it's pumping it against the concentration gradient, it is going to require energy because it's moving it where it doesn't want to go. So if we start up here, here's your membrane, here is this enzyme, the sodium potassium ATPase. It's going to be essentially this is your sodium potassium pump. That's what it is. So the yellow is the sodium. So it's higher concentration outside the cell and it's wanting to leak in. You don't want it in. So what do? You're going to right here pump. When you bind 3 sodium ions, that's going to trigger a shape change. And notice how this is like a gate. So you're going to close the internal section, open the outside. It just kind of flips it, kind of like on a hinge, releasing now the sodium outside the cell. Now, this turquoise color is your potassium. You're going to bind the potassium on. Once 2 are bound, it's going to clip, clip. That hinge goes back, moves the potassium inside. Now it's ready to pick up 3 more sodiums. So with your sodium-potassium pump, what happens is you are going to be moving 3 sodiums out and 2 potassiums in. Secondary active transport just depends on ion concentration. That's dependent on the primary one. And it's often used to help transport other solutes. So sometimes sodium can drag other molecules as it flows through the cell, like some sugars or amino acids with it. And this is just showing here how you've got some sodium helping, and it's bringing in some sugars in, some glucose in with it. So now that's how the sodium's in. How do you get rid of the sodium? Push it out by the sodium-potassium pump. So it's kind of like the sodium's coming into the cell by the front door, and you're just going to bring it out the back door. You will see the sodium-potassium pump come up again and again as we continue on studying A&P, because it is very, very important in helping to maintain this concentration gradient, which, like I said, is necessary for nerve impulses. It's necessary for muscle contractions. Once again, if you were to allow the sodium and potassium to reach equilibrium, same concentration inside and outside the cell, essentially, bottom line is the cell would die. And eventually, you would end up dying. Now, there's a size restriction as to what you can pass across that membrane. If it's too large, it's just not going in or going out. So how do you move very large molecules? Exocytosis means moving out of the cell. Remember, I know there's a lot of stuff that we're going to be studying throughout the semester. Break words down for the vocabulary to help you remember what it means. Exo means out. Cyto means cell. So exocytosis, you're moving substances out of the cell. Endocytosis, you're moving things into the cell. And there's a couple of different ways of doing this. Exocytosis, basically you form these little vesicles, these little sacs, and they're going to eventually move towards the plasma membrane and fuse with the membrane, and whatever was inside spills out. That vesicle then becomes part of the cell membrane. Probably one of the most common things we see are proteins that are made, and we'll get into a moment as to how they're made, but they then they get modified at the Golgi apparatus, and the vesicle breaks off from that and moves to the plasma membrane. In terms of moving things into the cell, phagocytosis technically means cell eating. It is where the cell is going to engulf material, and this is solid material. It's going to form what we call a vacuole, a little vesicle that has the food in it. It's going to fuse with what's known as a lysosome that contains a lot of digestive enzymes, and it's going to break down material and use what it can from it. So here it's engulfing this solid media, kind of wraps around it and engulfs it. This was part of the cell membrane. And now what it's going to do is it'll fuse with the lysosome. And here's kind of a cool picture showing food being ingested by a cell. Pinocytosis is very similar. However, now instead of solid material, it's ingesting fluid. So it's cell drinking. So phagocytosis and pinocytosis are non-specific. If there's food there, if there's fluid there, it'll engulf it. And here it is bringing in fluid. And this is a really neat picture showing the cell membrane with all of these vesicles forming here as it's engulfing material. Now, receptor-mediated endocytosis is very, very specific, and it's going to be dependent upon some of those receptor proteins that are embedded in the membrane. So what happens is when something binds with the receptor, that's going to trigger the membrane to kind of indent and form a vesicle that's containing that specific material. And I think it's best if you see here, it looks like a Y, the receptor protein, the red structure here, that's on the surface of the membrane. Well, here comes the specific molecule, the purple little triangle that binds with it. What about the green? It doesn't bind with it, so it's not going to do anything. When the purple binds with the receptor, it's going to trigger this infolding, and see how it kind of indents in and you form this vesicle. So now you've brought the little purple triangles inside the cell. So kind of in summary, you've got active transport and passive transport. Passive transport does not require energy. That's your diffusion. Osmosis, facilitated diffusion. Osmosis, you're just talking about water. Diffusion and facilitated diffusion, once again, all of those you are moving down their concentration gradient, so that's why it does not require energy. Facilitated diffusion needs some help of a transport protein. And then active transport, you're moving against the concentration gradient. This is where you have your ion pump, such as your sodium-potassium ion pump, and it's going to require energy. And then for large substances, if you're moving out of the cell, it's exocytosis. If you're moving into the cell, it's endocytosis. Phagocytosis is solid, pino is liquid, and receptor-mediated is very, very specific. We're going to introduce the membrane potential now. And once again, this will come back up when we talk about nerve impulses and muscle contraction. But the resting membrane potential, the RMP, basically what this is saying is because you've got your membrane that is separating things inside the cell from outside the cell. And a lot of these ions, by nature of their definition, they are charged. So a lot of these particles are charged, like your sodium, your potassium, calcium, etc. They have a charge to them. Well, what happens is you may have one charge on the inside of the cell and a different charge on the outside of the cell. And this is what we say is polarized. The cell becomes polarized. In a nutshell, what all of this is saying right here is that inside the cell, for most cells, it is going to have a slightly negative charge than outside the cell. And why is that? You tend to have potassium on the inside of the cell, which has a +1 charge. But sometimes you also have chlorine inside the cell. And outside the cell, you have a lot of sodium, which is a +1 charge. You have calcium, which is a +2 charge. So you have more plus charge outside the cell than what's inside the cell. So along the membrane, we can actually measure what those charges are and measure what the voltage is, the difference between the points. And depending on the cell, it ranges from -50 to -100 millivolts. One of the big players is going to be potassium in helping to determine what that resting membrane potential is. Potassium tends to diffuse out of the cell. There's some channels where it can leak out. So normally it's higher inside the cell, but remember your sodium-potassium pump is trying to bring it back in. Well, that +1 potassium charge is leaking out, but your negatively charged proteins can't leave. So they're stuck inside. So that's making it more and more negative. Now, with the sodium-potassium pump, you're trying to bring potassium back in, but it's still not equal. So the bottom line is, once again, most cells have a resting membrane potential around -90 millivolts. So now that the key player is potassium, know that most cells at resting potential is around minus 90 millivolts. What does that mean? That means that the inside is slightly negative. Sodium is going to play a role because it's on the outside. Yes, it's going to be attracted to the inside because opposite charges attract. So some cells, if sodium is able to enter, it's going to be around a minus 70 millivolts. So this is just showing here that you have all of this plus charge, a little bit of chlorine that doesn't really play that huge of a role, that's negative charge. But the outside of the cell is slightly positive, the inside is slightly negative. And like I say, this will come up again later when we're talking about nerve impulses and muscle contractions. So once again, here's your sodium potassium pump that's helping to maintain this now what we call electrochemical gradient. Is helping to maintain that positive charge outside, negative charge inside. And once again, it's 3 sodiums being pumped out, 2 potassiums being pumped back inside. And this pump, the sodium potassium pump, is constantly working, if you will. Every cell has thousands of different glycoprotein adhesion molecules that are sticking out from the membrane. What's the purpose of them? Well, to help to attach to the extracellular matrix or help cells attach to each other. It's helping to assist in cells moving past one another. When there is an injury, it will help to attract white blood cells to the site. It helps to stimulate both the synthesis or the degradation, depending on circumstances, of some of your junctions, like your tight junctions. And they can also help transmit signals to help with cell movement, cell specialization, etc. Some of these membrane receptor proteins serve as binding sites, remember, for other chemicals to come in. They can recognize each other. This is going to help with immunity, with normal development. Some of the chemical signaling can play a role in some of the chemical activities that are going to occur within a cell, whether it's activation of enzymes, whether it's going to open certain channels to allow things in or out, whether it sometimes like to allow hormones to bind. That's what makes the specificity. If they have a certain, say, receptor that the hormone can bind to, then that's a cell that will be affected by that particular hormone. If it doesn't have that receptor on it, then the hormone's not going to bind to it. It's not going to have an effect on it. So you can use the same response for several, or the same receptor for several different responses. Just depends on how the shape change occurs in terms of what is going to be activated. Sometimes activated receptors can open or close ion gates. So it's all directly linked to, once again, the proteins that are present in the membrane. That determines what type of processes, what type of reactions can occur with that particular cell. And that's basically what this is showing. Oftentimes we have what's known as a secondary messenger. On the outside of the cell, you have your first messenger. Here it's shown it binds to the receptor, which now over here, shape change, then activates this, which activates this. Over here, we get a shape change now. Over here, this big inside is your second messenger. It's just sitting around. How does it get activated? Because you have to have this first change here. So once again, it's this cascade type reaction, if you will. That's all looking at the membrane. Now we get to move to the cytoplasm. The cytoplasm is all that material that's inside the cell, but not in the nucleus. It's composed of cytosol, which is a solution. They used to say cytosol was mostly water. Well, it is made up of water. It also has things such as proteins, salts, and sugars in it. It's kind of almost like a gel-like solution. It is comprised also of inclusions. These are insoluble molecules. It depends on the type of cell you're looking at. There may be crystals, there may be pigments. It just depends on the specific cell. And then you've got your organelles. Your organelles, as I mentioned earlier, are structures that are inside the cell that have a very specialized function. We look at these organelles that are present in the cytoplasm. Some of them are membrane bound, some of them are not. The non-membrane bound ones are things such as your ribosomes, cytoskeleton, and centrioles. The membrane bound ones, which are only found in your eukaryotic cells, are things such as your mitochondria, the endoplasmic reticulum, Golgi apparatus, peroxisomes, and lysosomes. Now, an advantage of having the membranes bound ones, it allows for what we call compartmentalization. It allows for these specialized functions to occur, maybe in a slightly different environment than the rest of the cell, and that optimizes conditions for things to work out. One way of looking at it is, say if you look at my house, you have different rooms in your house. Different rooms have different functions. The function of the kitchen is very different than the function of your family room. Well, the environment might be very different as well for that. And so different environment allows for different processes to occur. I used to always say I have 3 kids, and we'll just say that my son's environment growing up in his room was very, very different than my youngest daughter in the environment in her room. It's the same type thing with the cell. The environment inside the mitochondria might be very different than that in the Golgi apparatus. Well, that allows for the different processes to occur there. So we start off with the mitochondria. The mitochondria is the powerhouse for the cell. Its function is to carry out cellular respiration, which produces ATP. So you're producing energy. It's converting that chemical energy in your food to the chemical energy that's used for process cell reactions. It does contain a double membrane. So if you look at the mitochondria, there is, because it has a double membrane, there's the intermembrane space. It's the space between the outer membrane and the inner membrane. And then the space inside the innermost membrane, so the very interior part of the mitochondria, is referred to as the mitochondrial matrix. And inside the mitochondrial matrix, there are some ribosomes, there are proteins, and there is some mitochondrial DNA. The inner membrane, it's not just a smooth line. It has all this infolding that we call the cristae. The purpose of this inner folding is to increase surface area. So whenever you see like any type of all these invaginations or infolding, that's increasing surface area, which tells you there must be something very important that occurs right there, which indeed that is where the ATP synthesis does occur. So if we look at this schematic drawing here, here's an electron micrograph. You can see, once again, there's the outer membrane, here's the inner membrane, all these infolding, that's the cristae. This inner membrane is where the ATP production occurs. Ribosomes, this is the site of where protein synthesis occurs. There's 2 different types of ribosomes. Free ribosomes, which are floating around, as the name implies, freely in the cytoplasm. These proteins made here typically are going to be used within the cell. And then your bound ribosomes are attached to usually the outside of the rough endoplasmic reticulum, and usually the proteins made on those bound ribosomes will be exported or secreted out of the cell. The endoplasmic reticulum, which is abbreviated as the ER, is this whole network of these tubes and sacs. It does start at the nuclear envelope and then extends out from that. There's two types, smooth and rough. Your smooth ER, the surface of it is smooth. There's nothing attached to it. And what is the purpose of it? Well, you've got, in most cells, lipid metabolism that will occur here. You may absorb, synthesize, or transport fats here. For some cells, that's kind of a detox center. Sometimes they will store calcium, such as in the skeleton and cardiac muscle cells. So not all of these processes will occur in all smoothies, or it may depend on the specific cell you're talking about. And then your rough ER does have the bound ribosomes attached to it, and so proteins are going to be made here. As I said, typically they will be exported out of the cell. Why would you make a protein just to export it out of the cell? Well, maybe it's enzymes that need to be used to break down very large molecules into smaller products that now you could easily pass into the cell. So from the rough ER, typically the vesicle is going to break off and then move to the Golgi apparatus. So this is showing, here's your nuclear envelope. So this would be inside the nucleus. Over here, no ribosomes attached. So that's a smooth ER. These little brown dots are the ribosomes. So this is your rough ER. And this is showing the rough ER with the ribosome attached to it. It is making a protein here. The new protein is passed from the ribosome inside the rough ER. It's going to fold up into its three-dimensional shape and be pinched off into this transport vesicle, kind of buds off, and now it's transporting it. So the protein is not directly in the cytosol. It's protected because it's inside the vesicle. The Golgi apparatus is where modifications to that protein you just made in the rough ER are going to be made. And so the vesicle is received on one side of it. It looks like a flattened stacks. I often say visualize it look like a stack of pancakes, and it's going to come in the vesicle from the rough ER. It's going to come in like on top, kind of like you're pouring syrup on the pancakes. So it's going to come on top. It's going to move all the way down. Now, the sacs are not interconnected. Just how you stack pancakes, one pancake is not directly intertwined with the one below it. While it's passing, the vesicle is passing through, it's going to be modified. So this is where you have modifications occurring on the protein. You may be sticking a sugar on it. You might be putting a lipid on it. And then the final transport vesicle is going to be taken. So majority of them, what's going to happen is pathway A, where the vesicle containing that modified protein is going then to be moved to the plasma membrane and exocytosis occurs. Sometimes if it's containing lipids, it may fuse with the plasma membrane and just insert whatever was just made directly into the membrane, if that was, say, repairing some damage. Lysosomes contain digestive enzymes, and they may hold the contents of the vesicle until it's needed. So this is showing here is that vesicle that came from the rough ER with the protein in it. So here's your Golgi apparatus here. You can see you've got these stacks of the sacs, or as I say, the pancakes. And then the vesicle comes in, it fuses. As it moves down, eventually it will be released on the other end. Modifications are complete now. Peroxisomes, these are used for breaking down fatty acids, breaking down hydrogen peroxide, which is H2O2, helping to detox alcohol and other various toxic compounds that have come into the cell. Your lysosomes, I mentioned these just a bit ago. These are a sac that contain highly digestive enzymes, so it's referred to as your recycling center. It will break down any food material. Some cells that are specialized to help, like with your immune system, in recognizing non-self, who are there to protect you, that tend to digest any bacteria or any other foreign substances. They tend to have a very high number of lysosomes in them. And so this is where you've got the plasma membrane. You have engulfed some material here. Here's a lysosome that contains those digestive enzymes. Your food vacuole now containing your food substances entirely within the cell. The lysosome comes over and fuses with it. Now the digestive enzymes can mix with the food, breaking it down, and the cell can use what it needs. There's a whole endomembrane system in the cell, starting with the nuclear envelope, which surrounds the nucleus. Your endoplasmic reticulum, you've already seen the rough ER and the smooth ER. The Golgi apparatus, your lysosomes, you've already seen that. Plasma membrane and vacuoles. These all work together. They're constant communication with each other. They're producing molecules, they're breaking down molecules, they may store molecules. If you don't need it, it exports it, it gets rid of dangerous compounds, all for the survival of the cell. So there's all this very coordinated work and processes that are constantly going on in the cell. Do you need to be consciously aware of all this? No, absolutely not. It'd be a little overwhelming it'd be a lot overwhelming for us if you knew what was going on at the cellular level. But you can see here with the nuclear membrane, it's continuous with the endoplasmic reticulum, which then transports to the Golgi apparatus. So there's got to be communication between all of these players. The cytoskeleton is this whole network of fibers or protein fibers that extend throughout the cell. They help to support the cell, provide that structural support and integrity, and they often are involved also with motility or movement of the cell itself. There's different types of fibers or filaments. There's the microfilaments. They are the thinnest, as the name implies. They do contain these strands of protein called actin. You'll see these with muscle contraction on how they are involved with that. They do assist with cell motility or movement of the cell, changes the shape of the cell. They are also involved in both endocytosis and exocytosis. The intermediate filaments, as the name implies, it's the intermediate between the microfilaments and microtubules. They do help the cell to resist different forces. And then the microtubules are the largest of the ones. They do help determine the overall shape of the cell. They do help with distributing where the various organelles are going to be held in place. Some of your motor proteins that are ones that will help function for movement, once again, of cells. So this just shows the microfilaments. Like I said, an example is the actin. You'll see these again with muscle contraction. The intermediate filaments, and then the microtubules. The centrosome and centrioles. Centrosome is located near the nucleus. It does contain the centrioles, which are, there's a pair of them. They're usually arranged at 90-degree or right angles to each other. They are involved with a couple things. They're going to be involved certainly with cell division. And then they can also help form the cilia and flagella. So this is what the centrioles are going to look like. Like I said, they're usually arranged at this 90-degree angle. This shows from a cross section. Now, some cells, not all, but some cells will have extensions. And they can be a couple of different types. They can be cilia or they can be flagella. Cilia tend to be short and very numerous. Flagella are much longer, and it's usually only 1 or 2 per cell. And microtubules are going to help them with movement. So these are extending out from the cell. So here's a picture of all of these little light color ones are cilia. They tend to move in unison, kind of like seaweed all moving together. An example of where you would find this is along the trachea, as you inhale. This is going to help move the phlegm and materials out of the lungs, prevent it hopefully from ever reaching the lungs, so they will move in unison. This is just showing the structure of the cilia. This is more just for your own information to see how it is related to the centriole down here, as you can see. And then this just shows how they will move kind of this back and forth motion. The idea is cilia, they are on a cell that is stationary, and it's going to move things across the surface of the cell. This increases the chances of the cell picking up nutrients, and oftentimes when all of these are working together in movement, it's going to move, as you can see here in this example, mucus along. And then flagella, like I say, is typically much, much longer than cilia, usually only one or two. This is an example of a sperm cell where it has just one flagella. Flagella helps a cell move through an environment. So cilia, the cell is still, and you're moving substances across the surface of the cell. That cell is stationary. Flagella is helping to move the cell itself through material. Microvilli, these are very tiny, small finger-like projections from the plasma membrane. Once again, they're not everywhere. An example where you would find this in the intestines, it helps to increase surface area for absorption. If you ate a lovely breakfast, you want to absorb as much of that nutrients as possible. So once again, this increases that surface area. And this is what it looks like. So instead of just a smooth surface here, with limited surface area, look how much you've increased the surface area now by having all these projections. The nucleus is the largest organelle that's going to be in the cell. It does contain the DNA, so it contains all of the control information, if you will, of the cell, how it responds to everything. Most cells only have one nucleus. There are a few that have more than one nucleus, such as some of your skeletal muscles, some of your liver cells. If it has more than one nucleus, it's called multinucleate. And then your mature red blood cells do not have a nucleus at all, so they are referred to as anucleate. There's three main structures of a nucleus. There is the nuclear envelope, the nucleolus, and then the chromatin. So this is showing the schematic drawing of the nucleus. So here's your nuclear envelope that separates the internal portion of the nucleus from the external portion. It does contain pores. So this whole thing is the nucleus. This is the nuclear envelope. Here's the nucleolus inside the nucleus, and then the chromatin, which is your DNA, is inside. The nucleolus, this is the site where ribosomal RNA synthesis occurs. Once rRNA is synthesized, it will be transported out of the nucleus. It's going to bind to certain proteins to help form your ribosomes. The nuclear envelope is a double membrane protecting the whole nucleus. Protects the DNA. It's just an extra layer of protection for it. It does contain these openings or pores that allow substances to move in and out of the nucleus, because if this is the control center, it needs to have communication abilities with the outside of the cell, so it knows what's going on and what it needs to do next. So the pores allow things to move in and out of the nucleus. And this is just showing the orientation of it once again. And this picture is, okay, for a science nerd, is really kind of cool because you can see actually some of these pores, which is really neat. The chromatin, this is essentially the DNA. The DNA is going to be wrapped around some proteins, which are known as histones. The DNA, we tend to use different terms. When the DNA, this long strand, when it's tightly condensed, so tightly coiled up around these histone proteins, we call it chromatin. When we stretch that strand out, then we call it chromosomes. And when the cell goes through cell division, it'll be in the chromosome form. All of it is DNA. It's just a way for us to distinguish what form the DNA is in. And so think of a ball of yarn. If it's all tightly coiled up, it's chromosome. If you pull that strand of yarn and stretch it out, excuse me, that's where it's going to be chromosome. The ball of yarn is going to be chromatin. So you can see here how the DNA, it's wrapped around, you're not seeing the first part of this where it's wrapped around the histones, which are proteins. But then you tightly coil this up and make these long strands and tightly coil it up till when it's tightly condensed, it is called the chromatin. DNA has been estimated to be about 2 meters, which is roughly 6 feet. And that's be tightly coiled up and fit inside your nucleus of your cell. Ultimately, DNA of eukaryotic cells is linear. I often describe it as it's kind of like when you get packaging, say at Christmas time, and you unpack it, and then you have to return the item, and it's like, there is no way this is fitting in that box. Yeah, your body has learned, it's amazing how it's able to take that long strand of DNA and tightly coil it up and fit it inside the nucleus to fit inside your cell. This is a very nice summary sheet of the various cellular organelles and what their function is. So it's kind of nice, it's all in one place. So it's a very good reference point for you. So once again, excuse me as I try to make these recordings with my cat who wants to be a part of it. The chromatin, like I said, is the DNA and the protein when it's in the long, thin fibers. You cannot see it with the unaided eye when it's in the chromatin form. The chromosome is when it is condensed, and you can see the chromosome form when you're looking at a microscope. DNA will be in the chromosome form during mitosis and meiosis, which are types of nuclear division. And then sister chromatids, I know we have a lot of vocabulary that sounds very, very similar. This is where you have started the process of DNA replication. So you had your original DNA, you've made a copy of it, and those two strands are still joined together at what's known as the centromere. So this is showing you have your DNA. So now, which is what the chromosome is, it went through DNA replication. The centromere is where it's the center point right here where each one of these are referred to as sister chromatids because one's the exact copy of the other. And the center, so think of them as identical twins if you want. They're still attached right here at the centromere. So that's where they're still attached. This is now the cell is going to go through cell division, but initially the DNA is still attached. The one copy is attached to the identical copy of it. So that's what you call the chromatids. They will eventually separate. So when we look at the life cycle of a cell, of course we're going to divide it into different parts. First, two parts of cell cycle are interphase and the mitotic phase. Interphase is the portion of the cell life cycle where the cell's going to spend most of its time. So a brand new cell, newly formed cell, will initially start off in interphase. It's going to be growing. It's carrying out all kinds of chemical reactions. Like I said, this is where it will spend most of its time. At the end of a cell life cycle, it will pass through the mitotic phase, which is when cell division occurs. And you will go from one cell to two cells. So an interphase, as I just said, most of the time is going to be spent here. It's a period where you have a lot of metabolic activity. And what do I mean by that? You have got all kinds of chemical reactions occurring. You're making proteins, you're breaking down sugars. Exactly which reactions are occurring, it depends on what the cell is, what the needs are at that particular time, which might change in 10 minutes. Interphase is divided into three small subdivisions: the G1, the S, and the G2. The G1 stands for the first gap. This is where you have a brand new cell enters into that G1. You have a lot of cell growth occurring. The S phase, the most important thing that occurs here is this is when DNA replication occurs. If ultimately you're going to go through cell division, each brand new cell needs to have its own copy of the DNA, which means you had to make that copy before you go through cell division. So this is when DNA replication occurs. And then G2 is the second gap. This is where cell growth continues, and you're going to be preparing for a cell division. And then the mitotic phase, which often is abbreviated just as the M phase, this is also subdivided into two parts: mitosis, which is when the chromosomes divide, and then cytokinesis, which is when the cytoplasm will divide. So you start here with a brand new cell. The green area here is interphase. As you can see, it is subdivided into G1, S, and G2. And then the mitotic phase, or the M phase, is subdivided into mitosis and cytokinesis. At the end of this, you'll end up with two new cells, which then start through this whole process again. So we're going to first start with DNA replication that has to occur during that S phase. The original parental DNA strands. DNA exists as a double helix, so those strands have to separate. And each one of those strands now becomes the template to make daughter strands. It is dependent on the specific base pairing that occurs. DNA replication is known as semi-conservative. So what this means is that the DNA is going to separate. You're going to use each strand as a template. Do you end up with the two original strands going back together and the two brand new strands going together? No, you do not. What happens is it's semi-conservative, meaning that you end up with one original parent strand and one of the new daughter strands. So you have your original parental DNA right here. It's going to separate. The gray is showing how you are adding in your nucleotides. And remember complementary base pairing. And yes, you do have to remember things from previous chapters. It all comes together. Remember, A and T always bind together, and C and G always binds together. So when you opened up and revealed the original parent strand as a template, what happens is that's used as a template. So it sees, oh, here's an A, I must put a T. Here's a C, I have to put a G. Here's a G, I put a C. Here's an A, I'm going to put a T. Here's a T, I'll put an A. Same thing on this strand. You're putting in the complementary bases. And so what do you end up with? The blue is the original parent strand. The gray is the new daughter strand. So you end up with one original and one new. That's semi-conservative. And this is shown here just from a different viewpoint of how you can see the double helix. It has to unwind, and then you're making the new strands along with it. So how does DNA replication itself actually occur? First off, you have what's known as origin of replication. These are certain sites where proteins can attach to the DNA, and they're going to ultimately separate the DNA strands. you have to untwist that helix first. You have to relieve the stress and untwist it and then allow it to separate. Now, from this point of origin, this point of replication, can go by either name, replication is going to occur in both directions from that point. And in eukaryotic cells, you have several origin of replication sites. So you can have multiple sites of where DNA replication is occurring. So what does this look like? Hopefully this clarifies it. These little black dots here are your origin of replication sites. So you have multiple ones of these. That is something that is different from prokaryotic cells. Prokaryotic cells like bacteria only have one site, but eukaryotic cells, animal cells like human cells, will have multiple ones. So you can start here. That's where it's going to open up. You can see the gray line. That is the new strand, new daughter strand. The blue is the original parent strand. But it's occurring in the DNA replication is occurring in both directions from that origin of replication site. And we actually call these replication bubbles. We give it a logical name. And they continue. As you can see, they're getting larger and larger until they merge together until you end up with two new strands. DNA polymerase is an enzyme that is involved with DNA replication. It's necessary for it. It is able to create the new strand by adding the appropriate bases. It can only work in one direction, though. It can only add on to what's known as the 3' end. And this has to do with structurally when you look at the sugar. Remember, a nucleotide for DNA is you've got your phosphate group, your sugar group, and then your nitrogenous base. And so it has to do with the orientation of the sugar. Well, it can only add on the 3' end, so the strand grows only basically in one direction. DNA, remember, the strands are anti-parallel, so one strand's going in one direction, the other strand's going the opposite way. Because DNA polymerase can only read in one direction, it means that one strand is going to be made continuously, but the other one's going to be made in pieces that you then have to connect. So, as you can see here, the original parental, the same color coding, parental DNA is in blue. The new strand, the daughter strand, is going to be in gray. So, like I said, same color coding that you've seen throughout. So first you have to find that point of origin, open up, untwist. Now the parental strand is exposed to be used as a template. But remember the DNA polymerase, which is this large darker gray right here, this triangle-shaped molecule, that's the DNA polymerase. It can only read in one direction. So it has to read this way. So this one strand is made continuously. It can't read this direction. Okay, so what happens is as it opens up, it'll read here. Then it goes back. Oh, okay, now I can read here. Now as you open up here, it can read here. As you continue to open up this direction, it's going to come back here, and then we'll make a little piece here. So you're making all these little pieces. These little pieces of DNA, these little fragments are called Okazaki fragments. They need to be connected together, which they will be by this other enzyme that's known as DNA ligase. I often say it's kind of like your DNA superglue. You've got to connect right there. When the DNA polymerase here moves and starts over here on the next section, the DNA ligase will come here and connect those sections together. Now the DNA polymerase can also act as a proofreader, so it moves along. If it sees it's put the wrong base in, it's not the complementary base, it will remove it and then add the correct base. So it's just kind of, it's booking it. It's just reading that original parent strand, using that as a template, going, oh, here's an A, let me put a T. There's a G, I put a C. Here's a C, put a G. Here's a C, put a G. Here's an A, put a T. Here's an A, put a T. Here's a C, put an A. Oh, wait a minute. That's the wrong one. Back up, take that out, and put the correct base in. They have been able to estimate that DNA polymerase is adding bases at a rate of about 2,000 bases per second. So it is moving. So here's another picture that has it all in. It's a summary sheet. Hopefully this will help you to see that you have your original DNA over here. You have your the point of origin was back here, but as you move along, you've got your helicases that have to untwist the DNA, open it up, so you're moving in from right to left. You're using the exposed bases on that parental strand as a template to add in the appropriate new bases here. Here's your DNA polymerase. As a new strand is being made, it twists up once it's done into double helix. And then this would be your lagging strand made in pieces. When this strand comes along, makes that section. By now this part has opened up. It's going to move over here and then do this. And like I said, the DNA ligase comes in and connects all those fragments together. So that all occurs during the S phase. Now your mitonic phase is divided into, remember, mitosis and cytokinesis. They are technically two separate events. Mitosis is when your chromosomes will divide, and then cytokinesis is when the cytoplasm division occurs. Now they are timed to end at the same time, but they technically are two separate events. These stages of mitosis, it is divided into 4 stages: prophase, metaphase, anaphase, and telophase. There are different ways of trying to remember this. Some people know it as PMAT. One way that I often teach it that helps college students remember it is pass me another tequila. Use whatever works for you. So what's occurring in all these different phases? Well, in prophase, the chromatin, remember that DNA, was kind of loosely stretched out. It's going to tightly condense up. Think of your yarn tightly going up into the ball of yarn, the skein of yarn now, into what's known as the chromosome. You do have 2 sister chromatids. Remember, DNA replication has already occurred, but those sister chromatids are attached to each other. The nucleolus is going to disappear. Your nuclear membrane is going to disappear. The centrosomes that have the centrioles inside, they start to move away from each other. They're going to start moving to opposite sides of the cell. And spindle fibers will begin to form. Where are they forming? From the centrioles. And they are going to attach to a protein that's known as the kinetochore. And we do have pictures of this in a moment. In metaphase, the kinetochore is in the middle where of the chromatids, the 2 sister chromatids, in that centromere where they're still attached. That's where the kinetochore is. So a spindle fiber will attach to the chromatin on one end, and the other end is attached to the centrioles. In metaphase, think middle. The centrioles have now moved to opposite ends of the cell. And then the chromosomes are all lined up right down the middle in what we call the equatorial plate. Anaphase, think apart. The sister chromatids now are going to separate. How are they going to separate? Because the spindle fibers are going to contract and be pulling them apart, essentially. The cell's going to elongate a little bit. And then telophase is essentially the opposite of prophase. The nuclear envelope reappears, the nucleolus reappears. Those chromosomes that were tightly coiled up, your ball of yarn, it's going to uncoil it back to the chromatin form, the long stretched out form. The spindle fibers are also going to disappear. Cytokinesis, as I said, is technically a separate event from mitosis, but it is timed so that it will end at the same time that telophase ends. It's the division of the cytoplasm. So when mitosis and cytokinesis have completed, what do you have? You will have 2 new daughter cells They are identical to each other. And when I say identical, I mean genetically identical to each other, and they are genetically identical to that original parent cell. So this is showing here interphase technically is not a part of mitosis, but this is where the cell was growing. You can see here you've got your nucleus with the chromatin in it. Here's your nucleolus. Move into prophase, and you start to see the nucleolus has disappeared. You notice the nuclear membrane's starting to disappear. Over here it has finally completely disappeared. Notice the DNA is tightly coiled up into the chromosome form. The DNA, you can see when it's in this form under just using a regular light microscope. You can see it. You'll notice the spindle fibers are forming. Centrioles that were together here in interphase are moving apart until they get to opposite ends of the pole. And the spindle fibers, this is the kinetochore I was talking about right here. It's a protein that's right in the middle, basically where the sister chromatids are attached. So you've got a spindle fiber attached on one side and a spindle fiber attached on the other side. When they are all lined up, the chromosomes all lined up right down the middle in the often referred to as the equatorial plate, but it's all lined up right down the middle. This is metaphase. That's why I said think metaphase, middle. Anaphase, think apart. What happens is these spindle fibers are contracting, so if they're connected to the kinetochore here, it's going to pull it. Think of tug-of-war. You're separating them. So the chromosomes are being pulled. All of these are being pulled to this centriole. All of these are being pulled to this centriole. And then telophase, you'll notice you're starting to see the reformation of a nuclear envelope. You start to see the reformation of the nucleolus. Spindle fibers are disappearing. The DNA is starting to relax and stretch out into that linear chromatin form again. Like I said, it is timed to occur with cytokinesis, which is the cytoplasmic division. So you see this pinching in, if you will. Ultimately, you'll form two new daughter cells. So, once again, back to this original diagram, you have a brand new cell. It enters into the G1 phase. Now, these checkpoints are different checkpoints along the way to make sure everything is progressing the way it is supposed to. If there's a problem, it will not be allowed to progress any further. DNA replication occurs in the S phase. The G2 phase, you're continuing to prepare for cell division. There's a major checkpoint right here before you enter into the mitotic phase. If for some reason DNA replication did not occur properly, then it would not be allowed to continue into mitosis. And through mitosis, there's also another checkpoint. So there's several control mechanisms trying to assure that all this proceeds the way it is supposed to. And this is a picture. Now, this is actually with plant cells, but as you can see, it's kind of neat to see these lighter colored ones. This is cells that are in interphase. You can see the nucleolus right there. And then when it's darker like this, that is in prophase, where you can see the chromosome form. The DNA is tightly coiled up. This is also prophase over here. This one right here is metaphase. You can see the chromosomes all lined up. This is a good example of anaphase where they're being pulled apart. And over here is telophase. When they've reached opposite ends, you're starting to shortly see, and also right here is another excellent example of telophase. Here you can see that the membrane's starting to form around the nuclear membrane again, until you eventually end up with two new cells. The timing of the cell division has to be very, very tightly controlled. You want cells to only divide when they are stimulated, so that you can control this. You don't want them just randomly dividing. Some cells will never divide. Usually they're stimulated by growth factors. There's a growth hormone that releases these growth factors that do stimulate different cells to divide. So as I said, there are different control mechanisms for this. There are certain other factors besides the growth hormones that will play a role. There is a certain surface to volume ratio cell. The cells can only get so large. They can only take up so much space. There are some stop signals that include availability of space. Normally, cells will stop dividing when they come in contact with other cells. It's known as contact inhibition. You cut yourself. You need to have the cells go through mitosis to repair that damage. But let's say you have a cut. You don't want the cells to just continuously grow and grow and grow and you end up with this huge growth, say, on your arm. You just want it to fill in the area of where you've had the damage. And that's where the contact inhibition comes in. So that once cells are touching each other, typically they will stop cell division. Now, there are times when these control mechanisms do not function properly and cells are able to grow without the regulatory controls in place, and that's when you end up with cancer. The size of the cell is going to be limited as to how big an individual cell gets because nutrients, gases have to diffuse in, waste products have to diffuse out of a cell. And so you have to have enough surface area for these substances to move in and out, but you can't have it too large. So if you look at a specified area, specified volume, if you have just one cell in this space, it's going to be hard to get what's required into the interiormost portion of the cell. Isn't it much more efficient in that same volume to fill it with smaller cells that easily you can have things diffuse into the middle here versus here? You're also increasing the amount of surface area. So therefore, actually, you can get more things in, have more products being produced. It's much more efficient. One way of thinking of this, let's say that we had a large auditorium. And if we had, let's say in a big class, let's say we had 30 people in a class, big auditorium classroom. If I were to bring in pizza, let's say I brought in, I don't know, let's say 5 pizzas. If I brought all 5 pizzas in to this classroom, so think of this as a classroom. So if I brought this in and I had 5 pizzas, if I brought all 5 pizzas and put them right here, and people are seated throughout the room, but I put all 5 right here, it's going to take a while for them to get back here. And good luck to the person back here. I hope you get some pizza. Wouldn't it make more sense to divide it into much smaller components? In other words, put a pizza here, put one here, one here, one here, and one here. It's much more efficient that way. Now, as we continue talking about the cell, and this is going to relate to different processes going on in the cell, you have RNA, ribonucleic acid, one of your types of nucleic acids. There's 3 subtypes of these that we talk about: messenger RNA or mRNA, transfer RNA, referred to as tRNA, and ribosomal RNA or rRNA. Now, your messenger RNA is going to be used in translation. It's going to be made using DNA as a template, and then this messenger RNA then itself will be used as a template for protein synthesis. Protein synthesis essentially is translation. Your tRNA or transfer RNA is going to be involved in protein synthesis or translation in that it is going to be transporting the amino acids to the ribosome. You know that you make proteins at the ribosomes. You know that proteins are a sequence of amino acids. So that means you have to get the amino acids to the ribosome. Who's going to do that? Well, that's the job of the transfer RNA. And then the ribosomal RNA. This is a structural component, as we've said earlier, of the ribosome. It's made in the nucleolus, it's transported out into the cell, combines with proteins. So your ribosome is structurally made up of proteins and RNA. Now this is just showing a kind of a summary of the processes that we're going to be talking about. So we are going to use DNA as a template to make RNA, and then RNA is used as a template to make proteins. So the process of DNA used as a template to make RNA is known as transcription. And then the process of using RNA as a template to make protein is known as translation. I know that people often will confuse these two terms, transcription and translation. Think of it this way. When you transcribe something, you're simply copying it. So you're going to take the DNA and you're going to copy it for RNA. They speak the same language. They're both nucleic acids. Their building blocks are the same. Now, from RNA to proteins, think of it as they speak a different language. Their building blocks are different. Building blocks of DNA and RNA are your nucleotides, but the building blocks of protein are amino acids. So you're going to have to change your language here. You can still use RNA as a template, but you're going to have, think of it as you're going to have a translation dictionary there to help you. So transcription means that you're just copying. Translation means you're changing languages. So it's as though the DNA has a message. Hi, how are you? And then RNA receives that message. Hi, how are you? But now it has to transfer that message down here, but it's got to change the language now. So it's going to take that message of hi, how are you, to say, oh, bonjour, comment allez-vous? So it's got to change the language. That's what translation is. With our eukaryotic cells, DNA, in that process of transcription and making RNA, that occurs in the nucleus, which makes sense because where's the DNA? DNA is in the nucleus, so it's going to have to occur in the nucleus. But if you're going to make proteins, the ribosomes are in the cytoplasm, which means you're going to have to move this RNA out into the cytoplasm before translation can occur. So the location is different, and this is true for eukaryotic cells. This is not true for prokaryotic cells, just to let you know, because they don't have a nucleus. So what's the process of transcription? In transcription, we're using that DNA as a template. We are only going to use one strand. We're going to ignore the other strand. Yes, I know DNA has two strands, but it's only going to use one of them. And it's only going to use a portion of one. It's not going to read the whole thing at one time. It's going to make that messenger mRNA. Once again, location for this for eukaryotic cells is in the nucleus. For prokaryotic cells, it would be in the cytoplasm because they don't have the nucleus. So what are you going to do? Your original strand of DNA is the blue. And we're going to say this section, this upper portion, the darker blue, is the strand that we are going to use as a template. Excuse me. So what happens? We're going to start complementary base pairing states where you see a T, you're going to put an A. Where you have an A, remember we're making RNA. T is found only in DNA. So the complementary base becomes U for uracil. Uracil is only found in RNA. You have a C here, so you're going to put a G. T, A, T, A, and all the way down until you reach an area that says, stop, this is all we need to read. Complementary base pairing rule. Now, the promoter area, this is the area on the DNA, and it's your starting site. It's where RNA polymerase is the enzyme that's going to come in and it will bind to the promoter area, and it's going to place that RNA polymerase in the proper orientation. It's going to help determine which of the DNA strands is the one you're supposed to read. And then the RNA polymerase is very similar to DNA polymerase in that it's going to place the correct complementary base, forming that new mRNA strand. So this is showing you've got to, number one, have that promoter area. Your RNA polymerase is going to come in. It's going to read in one direction. You've got a bubble here that's opening up as you move down. So it's going to be opening up this way. It's going to be closing the DNA up back behind it. DNA wants to be tightly coiled up in that double helix. It does not want the strands separated. So it's easier to see here. This little green area is the promoter area. So that's the starting section. Here's your RNA polymerase. Is it going to bind over here? No, it's only going to bind here. All right. Binds and it's going to orient it. You need to go this way, left to right. That's the way you got to go this time. So it's going to start reading as it moves along. So the initiation is binding to the promoter site. As it moves along, it's reading that DNA template strand, putting in the complementary base pairings, making that RNA strand. As you can see, the pink here as it's growing longer and longer as it proceeds down, and it continues until it reaches this stop codon. And then the RNA polymerase will detach. The mRNA that you just made detaches from the polymerase. The DNA has closed back up. So in eukaryotic cells, once again, it occurs in the nucleus. Now your mRNA is going to have to be modified before it can leave the nucleus into the cytoplasm. There's going to be some changes that have to occur. So the initial steps are initiation, that's binding to the promoter area. Elongation is just using that complementary base pairing. And then termination is when it reaches the stop codon and it dissociates. But now you have to do these modifications. The first thing is you have to add some extra nucleotides to the end. You have to add a cap, which is a guanine on one end, usually just one. And then you have to add what we call the tail, which is anywhere from 50 to 250 adenines on the other end. So you're adding nucleotides on either end of the mRNA. Why are you doing this? Well, it's going to help to transport the mRNA from the nucleus into the cytoplasm. It's going to help protect it once it's in the cytoplasm from all these various enzymes that are present. And it's going to help it basically to bind ribosomes. So your protein synthesis can occur. Now there's some other modifications that also have to occur once you've added the cap and the tail. With eukaryotic, and this only occurs in eukaryotic cells, just so you know. You just read the DNA and copy for the segment that you were told from point A to point B, which is from promoter to terminal. You read the whole thing of the DNA. Now you may not need all of that DNA. There are certain regions within there that we call non-coding regions. You don't need those, and those are called introns. The areas that you do need are called the exons. Well, if you don't need the introns, you have to get rid of them. And so you're going to be removing the introns and then joining the remaining pieces together, and that's called RNA splicing. Once that is done, then your mRNA is considered mature and it can leave the nucleus. It's kind of like, okay, now you've grown up, you can leave home. So up here at the top is your original DNA strand. So you read from the promoter to the terminus. This is your mRNA, this pink, you read everything. For this particular protein that you got the message that you needed to make, that it's needed right now, is embedded in here in the darker pink. But you don't need the light pink. Those are the introns. So you're going to cut those out, and then you're going to splice these together. How do you connect these together? Use DNA ligase. It'll help glue them together. And then now you've got this much shorter piece of mRNA, which is now considered mature, and through the nuclear pore, it's going to leave and go into the cytoplasm. So your mRNA contains a sequence of bases, but the protein is a sequence of amino acids. So that means you have to convert from bases to amino acids. This is where you have to do your translation. So each gene has a very specific base sequence to it. What your translation code is what we call a triplet code. It's known as a codon. Every 3 bases excuse me, is known as a codon. And 1 codon is an amino acid. So every 3 bases is essentially 1 amino acid. The genetic code, number 1, is universal. It's the same whether you're talking about animal cells, plant cells, bacteria cells. It's all the same. And what it is, it's a set of rules that we've found that shows this relationship between the codons and the amino acids. There's 24 codons. There's only 20 amino acids. So that means there is some redundancy here. And like I said, it is universal. So it doesn't matter what type of cell, this holds true. So things like, to give you an example, AUG, adenine, uracil, guanine, those 3 bases in that particular sequence is known as the start codon. It actually codes for the amino acid methionine, but that is always the start codon. There are 3 stop codons which do not code for any amino acid at all. And those are UAA, UAG, and UGA. With the codons, there's also what's known as a wobble effect, which I'll show you in just a second. So this is showing a table of all the codons, all 64 codons. And when we say there's redundancy, this is what I mean. Look here, you've got this is showing that the first nucleotide base is going to be C, the cytosine. Then uracil is the second one. You have several of them, and even continuing up here, there's actually 6 codons for leucine. That's because leucine is a very common amino acid. Serine, AGU, is for serine. Here is the start codon, and these are the stop codons, which do not code for any amino acid whatsoever. The wobble theory just shows what that states, and not all books. So your genetic books will talk about it, but not all general biology, just so you know. Look here at the 3rd position of the codon. The first 2 are the same, but the 3rd position, and this doesn't always hold true, but in this case for leucine, it's not as critical, and that's why they call it the wobble. If it was supposed to be C and you had a mutation and you stuck A in instead, you're still going to end up with the same amino acid. So it's not as critical. Now, you change the first position here for the first base. Oh, that's going to be devastating, obviously. You usually end up with a totally different amino acid. But the 3rd place, now sometimes here, like you see, the 3rd one, yes, it will make a difference. But oftentimes it does not. But those are your 64 codons that code for, like I say, every 3 bases code for an amino acid. So on this side, this is the amino acid. So you've got like glycine, serine, threonine, alanine, valine. These are all the amino acids. So there's 20 of them. tRNA, the transfer RNA, obviously is going to be involved with translation. It's going to pick up an amino acid that's floating around in the cytoplasm, and it's going to bring it to the ribosomes where the mRNA is, and it can recognize a codon on the mRNA and bind in the appropriate spot. And I'm going to walk through this in a moment, and so this will make sense. tRNA is single-stranded, as most of your RNA is. It's looped at one end. That's called the anticodon region. That is where The anticodon region is going to be complementary to the codon, so it will bind to it. Completely opposite from the anticodon region is your amino acid binding region. So that's where the amino acid is going to be bound. Now, it's very, very specific. If it binds to the amino acid serine, that is the only amino acid it will bind to. So obviously you're going to have lots of tRNAs because you've got 20 different amino acids. So this is one way of drawing what the tRNA looks like, where you have an anticodon region, whether you look at this diagram or over here. You have the anticodon region. This is the area that's going to bind to the mRNA, and this is the area that's going to bind to an amino acid. So your ribosomes, it's a site of translation to occur. Ribosome is composed of two subunits. Eukaryotic cells have two subunits, prokaryotic cells have two subunits, but the sizes of those subunits are different. Prokaryotic cells, their ribosomal subunits are slightly smaller than the eukaryotic ones. These subunits are composed of both proteins and your rRNA, the ribosomal RNA that was made in the nucleolus. So the ribosomes, I'm sure now you're starting to, okay, back up. She's talking about protein synthesis. So you've got your mRNA that has the template. The site is going to be the ribosomes, which is made up of two small subunits, but she's also talking about tRNAs. Okay. If the tRNAs are carrying the amino acids, you're wanting to make a protein, which is a sequence of amino acids. So you have to get two amino acids close enough together to form a peptide bond, to form a bond between them. Remember, a peptide bond is a covalent bond that is between two amino acids. Well, if you want to make a protein, that's a sequence of amino acids, you have to get all these amino acids close enough together to form those peptide bonds between each one. Do it one at a time. Add one amino acid at a time. Okay, well that's great. That means you have to get two tRNAs close enough together to form a peptide bond. How are you going to get them close enough together? You're going to bind them to the ribosome. The ribosome is going to hold them. It's like it's going to grab them, and I need you two to be right close to each other so we can form this bond. How do you know what sequence of amino acids you need for making your protein? That's where the mRNA comes in. It has that information on it. So this is showing a ribosome. You have a small subunit and you have a large subunit. The mRNA is going to bind right here between those two subunits. And the mRNA contains the information on how to make the protein, on what sequence of amino acids you must have. In the large subunit, you have these two compartments here that are binding sites. You're going to hold the tRNA here and hold another tRNA here. So you're going to hold them close enough together so you can form a peptide bond between the amino acids, which will be sticking off this end. Down here is the mRNA that holds the sequence. So the pink is the mRNA. Every three bases is a codon. Here's the anticodon, which binds to the codon region. The green is your tRNA. It's being held in place. You have another tRNA right here that's being held in its place, close enough together. The purple are your amino acids. So the amino acids right here are close enough together you can form a peptide bond. You already formed some here. So here's your growing chain. You're going to form a peptide bond right here between these two amino acids because you're holding them close enough together. That's the whole purpose of the ribosome, is to bring everybody together to make this process occur. So the steps of translation, you have initiation, elongation, and termination, just like you had for transcription. Elongation is going to be further subdivided into what we call codon recognition, peptide bond formation, and translocation. So initiation. Normally, the ribosome, those subunits, are separated. So you have to do a lot of bringing everybody together. You have to bring the mRNA in the proper orientation. You have to bring the two ribosomal subunits together, and you have to bring in that first tRNA with its amino acid. So the first step, the mRNA is going to bind to the small subunit. The tRNA with its first amino acid, which is always the start one, which is always methionine, it's going to come in and bind to the start codon. Now the large subunit is going to come in and bind. The tRNA is going to fit into what's known as the P site. So far, the other site, which is known as the A site, is empty. So to see this, here's your small subunit, here's your mRNA, and here comes the first tRNA. Now, if this was another tRNA, let's say, that was carrying the amino acid serine, well, its anticodon would be different. It would not be complementary to the AUG. So if it's CCC, it's not going to bind. It's just going to bounce off. This is the only one that's going to bind because it's the only one that has the complementary base coding. So once the tRNA has bound to the mRNA, which is bound to the small subunit, then the large subunit comes in and it places it in the P site. This is the P site, this is the A site. A site's empty. That is initiation. You've got everything set now. You're ready for elongation. This is where you're going to be adding one amino acid at a time. How do you know which one? Because the information is on the mRNA base sequence. Where did it get it from? It came from the DNA. The amino acids will be joined together by a peptide bond. So the first thing is you got to recognize the codon. So the anticodon on the tRNA has to pair with the complementary bases that are sitting now on the A site. The tRNA, when it comes in a bind, already has its amino acid that it picked up from the cytoplasm. As soon as it comes into the A site, the tRNA comes and binds. So the anticodon binds with the codon region, places it in the A site. That brings the amino acids close enough to form that peptide bond formation. Anytime you have a bond forming, remember you get a shape change. Along with the shape change, what happens is the tRNA is going to separate from the P site. And it basically is going to get kicked out. So now the P site's empty. Where does that tRNA go? It left its amino acid. It goes, picks up another amino acid. Remember, it's very specific. So if it had glycine, it's got to go get another glycine. The tRNA that was in the A site moves to the P site. Now the A site's empty to receive a new tRNA. This will continue until it reaches a stop codon, and that's going to be termination. There's no tRNA. So these three stop codons, there's no tRNA with a matching amino acid, and so the completed protein is going to be released. The ribosomal subunits come apart. The mRNA is released. Everything stops. So if we look at this, remember you had your tRNA in the P site. Here comes the new tRNA. If the anticodon region is complementary to the codon, it will bind. If not, it just bounces off and goes on its merry way. But in this case, it does recognize that it is complementary, so this one comes in and fills the A site. The amino acids are close enough to form a peptide bond. That causes a shape change. So you break the bond between the amino acid and this tRNA. See how it's released? It gets released from the P site. So this goes and gets a new amino acid. The tRNA in the A site moves to the P site. So it's moved over here to the P site. So now the A site is empty. And this just continues on and on. So tRNA comes in, peptide bond formed, P site, tRNA gets kicked out, P site's empty, A shifts to P. A is empty, new tRNA, form a peptide bond. Excuse me. P site's empty, gets kicked out, P site's empty, A moves to P. A is empty, new tRNA, bring in an amino acid, form a peptide bond. tRNA gets kicked out of P, P's empty, A shifts to P. A is empty, new tRNA, form a peptide bond. That kicks out the tRNA from P. P is empty, A shifts to P. A is empty, new tRNA. And this just keeps going round and round until it reaches that stop codon. Now on your mRNA, you can have multiple ribosomes attached to one single strand of mRNA so that you are making several different proteins. These would be the same proteins being made, but you're making several copies at once. It's much more efficient to do this way. One way, when I do face-to-face that I have you visualize this, is have fill up bags, but just to verbally describe it, if you had a project and we need to fill 50 bags with 6 cookies. If I had 6 people sitting at a table, each one has a different type of cookie, so each person has to put 1 cookie in the bag. If I were to, I grab a bag, hand it to the first person, and you put your cookie in, pass it to the second person? Do I wait until everyone has put all their cookies in, fold the bag up before I hand out the second bag? Wouldn't it be much more efficient to just get the bag, hand it to the first person, she puts a cookie in, passes it to the second person, he puts a cookie in. Meanwhile, I hand this bag number 2 to the first person. Don't wait till you have it all filled before you start the next bag. This is much more efficient. So the sequence of bases on the DNA, that information ultimately determines the sequence of amino acids in the protein. So genotype, that's what in genetics we refer to the information that is actually on the DNA. Phenotype is what you actually see. So genotype, information on the DNA, it produces or determines what the phenotype is. In other words, DNA determines what your physical traits are. What do you see? Do you see brown hair? Brown eyes? Do you see brown hair, blue eyes? What do you actually see? That information is on the DNA. So this is a nice summary sheet showing that transcription, which is occurring in the nucleus, your DNA is being used as a template here for making your mRNA. You make your modifications, move the mature mRNA out into the cytoplasm for translation now to occur. So transcription, remember, is using DNA as a template to make your mRNA. Translation is now using that mRNA as a template to make your protein. So it's protein synthesis. You have your steps of initiation where you have your first tRNA come in. Your mRNA is bound to the small subunit. The tRNA binds to it. Then the large subunit comes in and places that tRNA in the P site. Then you have elongation, where you are one by one adding amino acids, making that growing chain. You're making your protein. How does it know which amino acid to add? Because that information is on the mRNA, which came from the DNA. And this will continue until it reaches the termination site. Now, mutation is any change in the nucleotide sequence in the DNA. Not all mutations are bad. The location of it determines whether it's going to be harmful or not. This allows for rich diversity of the genes. So this is just showing once again the DNA, the process basically of transcription to translation, etc. The process where cellular debris is brought to the ribosomes for disposal, and the cell is very good at recycling. If it can't use it at all, it will dispose of it, but if it can recycle it, it will. It's very good about that. And apoptosis is programmed cell death. We do see this periodically where if a cell is damaged, it's going to go through this programmed cell death and then recycle any of the materials from it. Or if there's unnecessary cells, there's usually a series of steps that will release chemicals and it causes the cell to kind of shrink, and then macrophages will engulf it. Differentiation is the process of where cells, it's kind of like they get the information of how they're going to specialize. So all cells start off the same, and that's what we call stem cells. And then differentiation is where they get the information of, okay, you need to become a muscle cell, or you need to become a nerve cell, or you need to become a bone cell, whatever the case may be. So the DNA is the same in all of your cells. What helps with the differentiation process is on the DNA, which genes are turned on and which genes are turned off. So the DNA, say in your skin, is the same as the DNA in a muscle cell, but different areas of the DNA are turned on and off. That's how the differentiation process occurs. Hyperplasia is increased or faster cell growth. So that's one thing that happens oftentimes in cancers. You get this increased cell growth in the tumor areas, and atrophy is decreased or slower cell growth. There are certain processes that occur with cell aging as a natural thing. Part of it is kind of a wear and tear theory. The cell's exposed to more chemicals over time. The mitochondria may be damaged. Your system has been really actively working. It tends to weaken with age, and the cell division is going to slow and decline with age as well. So there's just part of these natural aging processes that we do see with cells. And some of it can be maybe exaggerated more with different environmental factors that it's being exposed to.